My guest today is Professor Timothy Cernak, who's Assistant Professor of Medicinal Chemistry at the University of Michigan. His lab studies the interface of chemical synthesis and computer science. Welcome, Tim.
Thank you. Thank you so much for having me, Gill.
Yeah, thanks for doing this. So I want to start with your 2021 paper, Reinforcing the Supply Chain for Umifenovir and Other Antiviral Drugs with Retrosynthetic Software. You say here the global disruption caused by the 2020 coronavirus pandemic stressed the supply chain of many products, including pharmaceuticals. Multiple drug repurposing studies for COVID-19 are now underway. It has been underway for a while now. If a winning therapeutic emerges, you say it's unlikely that the existing inventory of the medicine or even the chemical raw materials needed to synthesize it will be available in quantities required. So you say here we utilize retrosynthetic software to arrive at alternate chemical supply chains for the antiviral drugs, as well as 11 antiviral and anti-inflammatory drugs. So I was in a pharmaceutical company, Tim, a long time ago, and I was on the business side, so I wasn't a scientist. But I have a general idea what you're talking about here. And so when COVID-19 happened, what was the drug that was appeared to be a little effective? Remdesivir?
Yeah, remdesivir was definitely one of them that we looked at. Hydroxychloroquine is kind of an interesting one.
The famous hydroxychloroquine, yeah.
In the beginning days of the pandemic, right? Nobody knew, right? It was like, I mean, you could have someone on Twitter say, "I think hydroxychloroquine is great," and so it was a very interesting time. That paper was a very interesting scientific journey, I would say. That was easily the most intense period of my scientific career, because we started that on March 20th of 2020, when we shut our labs on March 13th, and then March 20th we started ramping back up for this work. And so it was there was just so much happening at that time in my life, both professionally and personally, and happening in the world around us at that moment. But it was an opportunity for us to pivot a research project that we were working on in partnership with Millipore Sigma. I should disclose that I do receive research funding from Millipore Sigma, and they market this Cynthia software that we used here. In the same way that we're currently seeing, there's like a shortage of computers and electric cars because there's not enough batteries to go in them, right? So there's like the parts that you need to build the product. And such is the case with pharmaceuticals, that, of course we could have the pills, we could run out of the pills, right? Rewind your mind back to 2020, to spring of 2020, and, it was like unclear what the new world was going to look like, right? I mean, like, a lot of us were running out of, cleaning supplies for our bathrooms and like dried pasta goods. So it was kind of it was an interesting time, and certainly the idea that like a sophisticated product like a pharmaceutical that would tackle this new alien virus, of course the supply of that could become quite limited. And so Millipore Sigma is a leading supplier of chemicals. They supply both the drugs for research purposes, but also the starting materials in quantity for the preparation of these drugs. And so they were quite concerned as a chemical supplier in spring of 2020. We don't know which starting materials we're going to get asked for. And because it was unclear which drug was going to be important, I mean, you in the popular media, we got to hear about hydroxychloroquine and remdesivir as repurposed drugs. Of course, now we get to enjoy the new drugs from Merck and Pfizer that are specifically targeted to SARS-CoV-2. But in the early days of the pandemic, it wasn't known which drugs were going to be important. And so at that time, in March of 2020, there were dozens of drugs that were undergoing clinical trials for repurposed drugs, right? So drugs that are already on the market for, say, diabetes or anti-infection of, non-SARS-CoV-2 type, maybe cancer, something, something not related to COVID-2, but they're biological agents. They're already approved by the FDA or other registration bodies, and so humans could get them quickly. But what if we run out, right? What if there's like so in March of 2020 there were only 5,000 doses of remdesivir, and like way more than 5,000 people who had COVID infection at that time. So we need more, right? We need chemical synthesis to bring us more. And so chemical synthesis is this really critical tool that I hope to talk about a lot today of where drugs come from, where everything comes from. It's like the actual making step.
So let me, from my own understanding, Tim, so we have a pharmaceutical process. At the end of that process, we get the pill, we get the fully encapsulated product. But there is a process with multiple steps, and at the beginning of that process, we have starting materials. And so if we need something in high quantities, the product at the end, we need high quantities of the raw materials. And if raw materials are not available, if I understand this correctly, Tim, you are essentially creating a different route to that product with a different starting material. Is that the way to understand it?
Yeah, that's spot on, Gill. I mean, we like to liken the process of chemical synthesis to Lego, right? And so, you piece the things, your Lego bits together, rather than the little nubbin fitting in the hole of a Lego piece, it's the formation of a chemical bond. And so the cool thing is that all day, every day, chemists are inventing new ways to make chemical bonds. There's new catalysts that will make these bonds. There's new reactions that will make these bonds. And so, a drug that was made in, say, the 1970s or 1980s, we could revisit that today with all the new catalysts and synthetic technology that exists today and find a completely new recipe to get there. We'd arrive at the exact same final product. They're the exact same molecule. They should have the exact same biological effect. But the starting materials that we use to get there are quite different. And so you can imagine in the case of a pandemic-induced supply chain shortage, that having an alternative recipe that allows you to start from different starting materials but arrive at the same product could be beneficial.
And so, sorry, Tim. So this could be a theme even post-pandemic, right? So this gives some flexibility to the supply chain, right? If we have multiple routes to get to the same product, it introduces a lot of flexibility to the supply chain. So even post-pandemic, this might be a way to think about drug manufacturing.
You're exactly right. The pandemic aspect of this and applying it to the supply chain of COVID possible drugs was obviously a pivot that not obviously, but it was a pivot from research we were already doing on improving synthetic efficiency and using data science as a tool to find alternative pathways to target molecules that we want to make. And so, I said that all day, every day, chemists are inventing new reactions to make chemical bonds. There's probably 100 reactions that came out today, right? If you read all the literature that exists, there's at least dozens of new reactions that came out today. It's not possible for a human to keep up with all of this literature. We have another body of work that tries to explore how many reactions there could be. And we don't have an answer to that question, but it's possible there's a near infinite number of possible reactions. And so it becomes this big data problem, and navigating through all of these reaction rules towards of course we want things to be made cheaply, we want them to be made safely, and so there's a lot at play here. And then we need to get back to a catalog of chemicals, like what can I actually buy in a bottle to start with. There's only there's about 50 million different chemicals you can buy, and which sounds like a lot, but frankly it's a very small number. There's an infinite number of molecules that we could make. So the big data problem emerges rapidly when you look at all the possible reactions you could run, all of the possible reagents you can buy, and all of the reaction conditions and catalysts you have to stitch these bonds together to get to a specific product that you want. We're very interested in how to make that process happen more effectively, and how reactions play into this. The reactions that we have can make the chemicals that we want, and we're also very interested in inventing new reactions. But my point is that there's lots of reactions, there's lots of starting materials, and it's a really big data challenge to try and stitch them all together and find your way through this morass.
Yeah, so you have a related paper from 2021, Ultrahigh-Throughput Experimentation for Information-Rich Chemical Synthesis. So you say here, the incorporation of data science is revolutionizing organic chemistry. It's becoming increasingly possible to predict reaction outcomes with accuracy, computationally plan new retrosynthetic routes to complex molecules. We talked about that, and design molecules with sophisticated functions. Critical to these developments has been statistical analysis of reaction data. For instance, with machine learning, yet there is very little reaction data available upon which to build models. Reaction data can be mined from the literature, as you mentioned, but experimental data tends to be reported in a text format that's difficult for computers to read. Compounding the issue, literature data are heavily biased toward productive reactions, and few negative reaction data points are reported, even though they're critical for training statistical models. So there are a few issues here, Tim. It's a very interesting and exciting problem. As you say, it's a big data problem. But computers don't understand text material, so you have to somehow translate that into a language computers could utilize to build statistical models or machine learning models. And machine learning models typically require both classes of data, both positive and negative classes of data. That's how machine learning models are trained. And so that is also an issue here, right? So it's almost like a cultural change is needed for the industry so that the data can be collected from both angles and can be collected in a form that could be utilized at scale, right? Is that the problem?
Yeah, you're exactly right. So there are a few things at play here. So let's just start with this discussion of positive and negative data. That, historically, when I was in graduate school, it was a faux pas to report your negative data, right? If you were to do that, your reviewers and your readers would say, "Oh, this reaction doesn't work," right? And so there's certainly a bias towards. It's still today it exists that I think that people are biased towards reporting the reactions that work in high yield to showcase, hey, my new catalyst, it works in high yield, right? And so it is important that we continue to push towards this high bar of performance where all reactions are working really well. We can always get a high yield, but that is, I'd say it's not consistent with reality right now. That I think any practicing chemist will tell you that for every working reaction, they ran like maybe 100, failed reactions, right? And so we do have this disconnect right now. It's being bridged in parts from the efforts of our community, but the reactions that exist in the literature are biased towards those that work. And so as you rightly point out, a machine learning model wants to look at a body of data and say, these are the criteria upon which the reaction works, and these are the criteria upon which the reaction doesn't work. And based on that, I can assign descriptors to the data, molecules in our instance, and say, okay, when a molecule looks like this, it should work. When a molecule looks like this, it probably won't work. But the machine learning models don't have that data available to them to train on, right? There's no, if as humans we have chosen to only publish the reactions that work, and the machine learning model wants to know what works and what doesn't work, where's it going to get that data? So here comes high throughput experimentation as a new tool where it's completely acceptable to publish what works and what doesn't work, and so we've been doing this many times over, where we in a high throughput format can run either a dozen or 100 or 1,000 reactions in a single experiment. We can, rather than saying, I'm going to take an electrophile and a nucleophile, think of these as like the two kind of bits of Lego that you want to click together to make your bond. As chemists, we always talk about electrophiles and nucleophiles, and they're the kind of Lego pieces that you bring together. So in a classic experiment, we might take a flask, we put in one electrophile and one nucleophile and see what happens. And if it works really well, then we get, maybe an 85% or 95% yield, and we put that in our paper. In a high throughput experiment, we use an array. Hopefully some of your readers or listeners will be familiar with 96 well plates. And so these are very common experimental tools that we use. And so now in a 96 well plate, you have an array, and you can put a whole bunch of nucleophiles on one axis and a whole bunch of nucleophiles on one axis and capture the whole system. And so, we just report that whether it works or not. Usually about half the plate works and half the plate doesn't. And then, of course, that gives us a really nice training set for machine learning models, because now we are reporting both what works, what didn't work. And so high throughput experimentation is a technology that allows that to happen, and something we've been building for a long time now.
So I don't know if it's possible, Tim, but if everybody can share their data from this high throughput experimentation, then it becomes very powerful. I don't know if there is an agency, there's some sort of an academic institution that can take a lead on creating sort of a global database where we get information from a variety of sources, and that database becomes very powerful in terms of figuring out what could be better, right?
I would say that we're right at the cusp of that happening. There's a few really beautiful efforts. The first that come to mind are the Open Reaction Database, or AutoQChem, our first kind of entries in this space. One of the things that is happening here is the birth of this technology is frankly in the pharmaceutical industry. And as you mentioned, I left Merck and Company 4 years ago to come to academia and do this. And so, it's relatively new in an academic setting. I would say that many companies are involved in high throughput experimentation, right? And so they recognize the power that this technology can bring to the invention of pharmaceuticals, the invention of materials and agrochemicals too. But, their remit is, on the one hand, every true pharmaceutical company has a remit to enhance the scientific good, but data can be proprietary. It's just difficult to find models to easily share this data. So I mean, what we are looking at is at the cusp of a revolution really in this high throughput technology. It's relatively new, and so there are definitely efforts to standardize the reporting of data. We are definitely involved. We are about to release for free academic use a software called Factor, which will standardize the format of reporting of all this data so that everyone can do it very easily. But the one problem that exists is that it's very difficult to get creative people to agree on exactly what a standard format looks like. My favorite analogy here is the 96 well plate. If some of your listeners are familiar with the 96 well plate or have seen one of these, it is the kind of the standard vessel for doing experimentation in the biochemical sciences, and we're trying to push its use in organic chemistry. But in the early days of 96 well, so the 96 well plate was invented in 1950. All right, so we've had this tool for over 70 years. But it was only relatively recently that there were standards for what a 96 well plate should look like. If you can imagine, for like 50 years, if you bought your 96 well plate from one supplier, it had a different shape and a different size than another 96 well plate. And so the beauty of 96 well plates is that you can take them from one instrument and pass them to the other instrument. It doesn't matter where they come from. That wasn't always the case, right? And it took a consortium of people to agree on what the standard dimensions of a 96 well plate must look like so that it could pass from instrument to instrument. It took dozens of chemists or scientists 16 years to agree on what that format should look like. And that's kind of where we are right now in terms of data reporting for reactions, because this kind of comes back to one of the earlier points that you brought up, is that the current format that's accepted is prose, is text. You just write your prose. We even try to be poetic in the way that we describe how we ran the experiment. And so, I think that has served the community very well for the past several decades. But now we're entering this new era where we want our computers to be able to read reaction data and to learn from them, to do machine learning on reaction data. And so prose is not appropriate anymore. And so it's just switching right now. Like in the past 2 to 3 years, it's happening right now.
I was also wondering, Tim, I don't know if it's possible. So once you have the high throughput equipment, so you have already taken the capital cost, you would rather, I would imagine, run it continuously and create data, right? It's just like a computer. You don't want to, let the computer sit idle if it can do something. Is it possible to think about it that way?
Yes, we're talking here about automation, and so long-term automation. I have high excitement for these types of projects. They are very expensive projects, or they can be very expensive projects. I happen to have the luxury of having graduate students and postdoctoral fellows working with me, and so I mean we've definitely scoped out, weighing out powders, right? So weighing out powders is like a very simple operation. It costs millions of dollars to automate this, to have a robot do this, because if you can imagine the diversity of chemical, I earlier said that there's about 50 million different chemicals you can buy. They have very different properties, right? Some of them are big chunky crystals. Some of them are like brick dust. Some of them are like, light fluffy powder. Some of them are like a wax. Some are like a honey. Some are oils. Some are syrups. And so it's just very difficult to have a robot that can handle all of this. Whereas a human can do it no problem, right? So we choose in my research program to automate what enhances our ability to be scientifically creative, but we don't try to automate what we can do readily by hand, unless it really gives us that that extra edge, or we do a lot of automation, but what you're describing, full automation, no human engagement, it is doable. I would say that we're on the, that I would call that a pretty cutting edge project. And so, we're very interested in inventing new reactions. I have PhD students who are all trying to do something very different, something very creative. And so the more that, one rule that I like to abide by is that, automation and flexibility are often at odds with each other. And so if you want to be very flexible in your science, it limits the degree to which you can automate. So we're very excited about automation. We definitely will set up 1,500 reactions and let a robot like analyze through those overnight. But we do, we try to really embrace the human's engagement in this process. We're not trying to automate away human creativity or human engagement. And I think that there's certainly an aspect of using your eyes and observing what's going on in the flask with your hands. That is just still quite difficult for a robot to handle.
Mm-hmm. Yeah, I was thinking more like a screening process. So given an objective function, given a requirement, and given a plethora of starting materials, the machine could select a dozen possibilities that I would imagine the human would ultimately make a decision on. So a lot of the, I don't want to call it grunt work, but a lot of the, repeated work could be delegated to the machine, I would think.
You're absolutely right. Yeah. So I mean, I don't want to give at all the impression that I dislike automation. I love automation. We're very engaged in automation. And exactly as you say, I mean, so this is one of our favorite things to do, is to focus on a particular area of reaction space. We want to make this type of bond. We want to use these types of catalysts. And I think what you'll find is that the number of parameters that's involved in a really focused objective like that will still give you millions or billions of reasonable experiments to try. So we very frequently will first map out that space and say, okay, here's our objective. This is the bond we are trying to make. And what are like the reasonable experiments we can do? And then how do we kind of chug our way through that? I think what we have found, though, is that it's often the case that we do 3 rounds of experimentation in that space, and the data that we collect changes our perspective so much on what, even though it might be set up to imagine 10 rounds of experimentation. By the time we get to the 3rd round, it's like, oh, there's all this stuff we hadn't even thought of that now needs to be incorporated, and we are already at millions or billions of experiments, and then so it just like rapidly grows.
And the machine learning aspect, so you could think about, I would imagine, reinforcement learning type mechanisms here too. So sort of a feedback coming back from the human to sort of direct, a comprehensive search of the design space, I think, would be very, very costly. But if you can direct the machine to favorable spots, then perhaps that process could get to a result a lot faster.
Absolutely. So this is happening. It's happening in our labs and in other labs. And I would say it's very much the cutting edge. Well, we have a paper that was recently released where we showed that the order of reagent addition was critical to the success of the reaction, and that's a little bit difficult to encode into your prior thinking. That I've talked about how we have an electrophile and nucleophile. We like to think of them as like a blue piece of Lego and a yellow piece of Lego, and you want to click those two together. So there's a reaction arrow that takes the two pieces of Lego and unites them into that chemical bond is formed. And above that reaction arrow, there's a whole bunch of stuff. So there's a temperature that we heat the reaction at. There's a time that we leave the reaction for. Sadly, we're learning that the rate that we stir the reaction with the magnetic stirrer is very important for most reactions. It's sad because it's hard to, want to think about that. And then there's very frequently there's a catalyst, there's a ligand that attaches to that catalyst, there's a co-catalyst, and there's a co-ligand that adds onto that. And then there's either an acid or a base. And a few other additives to kind of make the secret sauce work, and then there's a solvent that kind of dissolves it all and kind of makes it like a nice kind of bubbling purple liquid. There's an enormous amount of what we call reactivity cliffs in this space. That if you choose any one of those parameters wrong, you get nothing. And when you get nothing, it's a little bit like being out in the open ocean and trying to, we absolutely apply machine learning to these tasks. The bar is high for us because we're trying to discover new types of reactivity and explore new reactions that don't exist. In my prior life, I did a lot of work exploring classic reactions like the amide coupling, where you take an amine and an acid and you make an amide bond. And so that's like a well-known reaction. There's other like really classic ones like the Suzuki coupling. These are the Buchwald-Hartwig coupling are Nobel Prize winning or soon to be Nobel Prize winning reactions that are used all the time. And so we have when you're exploring in that space, it's not really like you're out in the deep ocean and you make a wrong step and you don't know where to go next. Like if you make a wrong step playing in those spaces, there's a lot that is known about the mechanism, about the reagents that work, about like what temperature regime you should be in, what types of solvents are okay for these reactions. So you have some like knowledge to go on. But for new reactions, I mentioned the amine acid coupling to make the amide bond is the most popular reaction in all of organic chemistry. We're trying to find new ways to click together amines and acids. And here we are frequently in a very unknown space. And so while we are interested in building out a big, reasonable experimental space and then automating our way through the navigation of that space, we're just very aware that we are out in the open ocean. And so oftentimes we need to do some very careful reconnaissance kind of cut our exploration short and go in a completely new direction because there are all these reactivity cliffs. You have your electrophile, you have your nucleophile, you have the reaction arrow to make the product, and you have a solvent and a temperature and a catalyst and a ligand and a base and a co-catalyst. And each one of those could be reasonably selected from the 50 million commercially available compounds. So you get this combinatorial problem where it's very easy to take a misstep. And so I think the thing that is not really well appreciated right now is that reaction space in organic chemistry is very bumpy. And so it's very easy to find, yourself skittering down a cliff where you're at a 0% yield. Coming back to what we were earlier talking about, right? It's like we don't talk about our 0% yields, but any practicing organic chemist knows that like all the reactions you run give a 0% yield, right? Almost no reactions really work. A few of them do, but it's a very cliffy place that we're walking around, and so it's very easy to make a very subtle change. You turn the temperature up 20 degrees and the yield goes down to 0%, and so we don't yet have the data lake available to us to make meaningful predictions out of nowhere. We're getting better. I wouldn't say we're even good yet, but we as a community are getting better at casting a net over a new area of reaction space or a well-known area of reaction space and then navigating through that using machine learning. But I still think that's very much the frontier of where we are as a field. And so we're learning how to predict how to invent reactions in that space. With I think the ultimate goal of the entire organic chemistry field right now is to invent reactions basically out of thin air.
Yeah, this is unrelated, Tim. This makes sense why we haven't been able to create a biological system from chemicals. It is not just combining things. It is actually a variety of permutations on top of it. Everything has to be perfect for something to emerge. And probably that's a reason life is quite rare, perhaps
Yeah,
even
so
in the universe.
you're bringing up the notion of the origin of life. I love it. Yeah, yeah. I mean, like how fragile a system, right? That just like, that like just the right amount of photons hit that, that geochemical catalyst at just the right moment, to bring together, the chemicals that became, I don't know, the first sugar or the first base pair or whatever. But how fragile it was, right? I mean, it took billions of years to for everything to line up just right. And I mean, we are thinking of reactions of about that complexity, right? Like basically we are interested in making sugars. We are interested in making heterocycles that look like adenine and guanine. And we're definitely interested in making amino acids. And so I mean, it's really interesting to hear you bring up the origin of life. And I mean the recipes that existed in prebiotic Earth of geochemicals, high intensity light, heat, pressure, and like pH gradients. That's what we're doing in the lab. We're basically we're trying to recreate it. But I would say I love what you've brought up here for me in thinking about the origin of life and how this comes together. I think we tend to think of it in a very industrialized way, right? It's like, society needs this type of bond, right? Like society needs more carbon-carbon bonds. And so we really want to focus on ways that we could, make carbon-carbon bonds that society needs. But nature is doing this all the time. Nature is so good at making carbon-carbon bonds. I wish we had a tiny glimpse of what nature can pull off in terms of making
No, I
carbon-carbon
was
bonds.
struck by, I don't know much about this, Tim. I was struck by what you said in terms of the temperature, even the stirring motion, everything has to be so perfect for something to emerge. Tells me that even if you have all the environmental factors right, you still may not get anything because some factor, some parameter has to be precisely right for that to happen. And that, I mean, I haven't really thought about it. People have been thinking about life emerging from a set of chemicals. That is not the case. It's sort of set of chemicals in a matrix of variety of factors that we haven't really even have a clue about. So that makes it a very, very challenging problem, I think.
Absolutely. I mean, so there's a lot of discussion around chemical space, like all the chemicals that could exist, and I've alluded to this a little bit. And then as practicing synthetic chemists, we like to think about all the recipes of reactions that could make those chemicals exist, and all those chemicals are they're going to help humanity in the future. They're going to improve our access to food, and certainly medicine I think is somewhere that enjoys a lot of engagement from the organic chemistry community. But so you can imagine a pill that a simple case would be a pill that you want to take once a day, right? Because I mean, I'm currently on antibiotics. I got a tick bite, and so I'm taking doxycycline right now. I have to take it twice a day. It's a pain in the ass. I cannot remember to do anything twice a day, right? So I don't want to do something like once in the morning, once at night. So I'd like to take one pill a day. But you can imagine another type of pill that you want to take once every hour, right? Or like not once every hour, but like a sleeping pill you want to take. You want to take it. You don't want that last all day, right? You'll sleep the day away. So you want a pill that lasts for like 4 hours. And so the difference between the 4-hour pill and the 1-day pill, or with doxycycline, I'm taking 8-hour, 12-hour pills, is very subtle changes in chemical space. And so I mean, this is, we're now getting into medicinal chemistry and pharmacokinetics, and I'm very excited about these technologies, or these fields as well. But you know, the difference between a 4-hour lifetime pill and a 24-hour lifetime pill is oftentimes just changing a carbon to a nitrogen. Right. And so this is chemical space. There's a huge amount of investment in chemical space because, of course, some of these single molecules in chemical space turn out to be multi-billion-dollar projects, and they more importantly save human lives. Right. So getting the atom placement just right can save a human life. And you know, give our customers, if we're thinking as a pharmaceutical company, a really enjoyable user experience. I'm frustrated that I have to take 2 pills a day, and so you know, maybe a pharmaceutical company could invent for me a once daily doxycycline dosing regimen. But all of this is chemical space, and this is well known. What we're doing that's a little bit different is looking at reaction condition space. And so you alluded to this, you know, this notion that like the stars have to align with just the right stirring rate and just the right temperature, and you have to pick just the right arrangement of atoms on your catalyst and on your base so that things work. So not only is there this infinite menu of products that we want to make that would be either the once daily pill or the 4-hour pill, that could come from, you know, the 50 million chemicals that we can buy, but there's the reaction that has to, you know, that turns these two things into this one thing, and forms that chemical bond. And it's a really rocky terrain that goes above that reaction arrow of you know, picking the right temperature, the right stirring rate, and all that. But I don't know, we're so excited about our opportunity to invent new chemical bonds. You know, from our favorite thing to do is to look at, you know, which of those 50 million chemicals are the most broadly distributed, and then find ways to make the chemical bonds that society needs most. Carbon-carbon bonds are a great one. So I mean, one of our main, you know, areas of interest right now is taking amines and acids, because there's so many commercially available amines and acids, and trying to find ways to make carbon-carbon bonds from them. And so we're having a lot of fun there. But we do a lot of experiments that fail. I mean, you know, if there were a journal that only reported 0% yields, we could publish in it every day, because we run lots of these experiments that don't work. We use our high throughput experimentation tools to, rather than run one experiment or you know, 2 experiments at a time, we'll run dozens to hundreds or thousands, and then we hope to find that kind of diamond in the rough, that by scanning more reaction experimental space at a time, we hope that we can find like a, you know, just that little peak on this really rocky terrain that will give us the reaction conditions we need.
Yeah, so I don't know much about it, Tim, but I was wondering, so this combinatorial explosion that you have in terms of finding something, I wondered underneath that if there is some sort of physics explanation. So empirically, you know, it just looks like such a huge design space. It's really difficult to navigate the design space. But there could be some physics that is more approachable underneath that, perhaps, I wonder.
Yeah. What you're onto something here, Gill. So yes, there is. There's so we call it density functional theory, or DFT is one of the tools that one would use here. Or well, the physics you're asking for is quantum mechanics. And so density functional theory, or DFT, is a way to do quantum mechanical calculations. So if you can recall, we don't have the computational power to solve the Schrodinger equation for hydrogen, right? So for just H bound to H, we can't do, right? So I mean, maybe once we have quantum computers, that's on the table, right? But right now we don't have the computational power to know exactly where the electrons are going to be on just hydrogen. Density functional theory allows us to approximate that, but it's still quite computationally expensive. And now we want to talk about remdesivir, right? Which is, you know, or Taxol or doxycycline. These are big molecules. They are much bigger than H2. And so solving the Schrodinger equation for such a molecule is you know, hopefully our grandchildren get to enjoy such technologies, but they're not yet available. But you highlight the physics are known, right? So we understand it's quantum mechanics that will explain all of the events of bonding can be broken down into this. But recall back to what I've been saying, that we have an electrophile, we have a nucleophile, we have a catalyst and a co-catalyst, and these things it's a Rube Goldberg machine. There's there's 18 different steps that our nucleophile and electrophile go through before they get to the product. And so we don't even draw it in the reaction typically, but there's a proton that's like shuttling around, and sometimes things are protonated, sometimes they're deprotonated. I talked about how we put a base into a reaction because sometimes the base needs to come in and like pull off the proton for one mechanistic step and then place it back on for the next one. And so it's really a miracle that reactions work at all. But as I told you, it's probably been dozens to 100 new reactions reported today, right? I mean, because chemists all around the world are very eager to find that new set of reaction conditions that works for all types of chemicals. And so so I've highlighted that chemical space is near infinite, and within there you can find your 4-hour pill or your 24-hour pill that hopefully we can find like the pill you only have to take once for your life, right? And then you are never going to get cancer, you're never going to get Alzheimer's, right? That is hopefully part of the future of medicine that will get there, and hopefully it's a molecule. It will be a molecule that can achieve this. But then there's 50 million building blocks we get to choose from. There will be more. Our grandkids will get to enjoy 50 billion building blocks. The largest collection of the building blocks in the world right now is in Kiev in the Ukraine, and so as chemists we're all very worried for this very important. Bless you. So there's all the building blocks we have. And then, I highlighted that there's many mechanistic reaction steps that one needs to go through to unite the nucleophile and the electrophile to make the product that will hopefully be the cure for Alzheimer's. And we need to pick just the right catalyst and temperature and stirring rate to make that happen. So it's the rules and the logic behind it are almost 2 centuries old. I mean, this field of chemical synthesis is almost 200 years old, but we're not yet there. So I mean, there's too much data, but machine learning is helping us for sure. I think anyone that tells you that chemistry is nearly solved and machine learning is like fix it all, they're totally lying to you. It's not going to happen in my lifetime. I believe that there's too much to explore, but yet machine learning helps us to quickly navigate focused areas of this experimental space, and so we're having a lot of fun exploring its role there.
Yeah, it's an exciting area. So I want to finish up with another really interesting paper from just recently, molecular sonification for molecule to music information transfer. You say organic chemical structures encode information about a molecule's atom and bond arrangement. The most established way to encode a molecular structure is through line drawing, although other representations based on graphs, strings, one-hot encoded labels, or fingerprint arrays are critical to the computational study of molecules. Here we show that music is a highly dimensional information storage medium that can be used to encode molecular structure. That is really interesting, Tim. So how do you use music to encode molecular structure?
Yeah, this one has been incredibly fun and is we opened quite a can of worms with this paper, and there's so much that we are currently exploring. The idea comes from having worked with these strings for the longest time. So hopefully you've got the impression that I'm interested in working with computers as I do chemistry. When I studied chemistry, it was all done at the chalkboard, right? You would go up to the chalkboard and draw in your atoms and the bonds between them. And I still love to do that. I have installed old school chalkboards in my labs because I think the chalkboard is a very important place to brainstorm and discuss. But so that's like one way that we interact with molecules. As we do all this machine learning research, we tend to use a special string representation called a smiles string. And so this is basically because the computer isn't a chalkboard, right? I mean, the computer wants to read text, but not prose that we talked about earlier. It's so smiles is a very specific way to talk about a molecular structure. And have the computer understand it. So I can type in a smile string into a computer, and it will draw for me the exact chemical structure that I would then go draw on the chalkboard. Not only will the computer draw it for me, but the computer understands it, right? The problem is that like as a chemist, I've been studying chemistry for a long time. I can't look at a smile string and really tell what it is, and I certainly can't like manipulate it in a way that I want, and have it turn into something new. And so, you know, we got to thinking about how music is a very tactile experience. Music is a very, you know, you have a keyboard, you have an instrument where you play it with your hands, you play it with your fingers, and you know, you manipulate it in a very tactile way. And so we, you know, we had spent so much time turning molecules into dots, molecules into, you know, graphs, and molecules into like histograms to do our analyses. We're like, what, you know, what if we could turn it into something that was more dimensional and something that was really creative? And so we take the information of a molecule and we turn it into musical information. I'll try to play for you something off my phone. I don't know if it'll work, but so is this coming through?
Yeah.
So this is the chemical structure of vitamin B12 turned into a jazzy little riff. And so we can feed this into our system and pull out the exact structure of vitamin B12. We can do this for many, you know, any molecule really is working. The beauty is that, you know, that you can create in this way. That, you know, we can go to the keyboard and we can jam out a tune and a new molecule will come out. The similarity of that melody to an existing molecule will determine how similar the new molecule is. So we can take a song of a molecule and we can change one note, and that will then somewhere in the molecule rip out a bond, or make a subtle change to it. We based this on a really fun algorithm called selfies from you know, my hero Alan Asparuh-Guzik and his incredible team up in Toronto. But we this allows us to generate new music or new molecules. And so, you know, it started as a question around molecules and information, and you know, how we're encoding molecular information. We do so much like just, you know, drawing scatter plots of molecules is very important for our research. We do that all the time. And so it's like, okay, here's one way that we're like, why are we looking at molecules as colored dots on a scatter plot? Why not listen to them as you know, information encoded music? So you can imagine we're having a lot of fun. One corner of the lab is totally filled with keyboards and DJ equipment and guitar pedals. We're exploring, you know, I mean, our dream is that, you know, exactly as we were talking about, I'm a medicinal chemist. I have a 4-hour pill and I want to turn it into a 24-hour pill, right? And so I'm going to go to the chalkboard and I'm going to take all of my knowledge in pharmacokinetics and in atom arrangements and in chemical synthesis and like erase this nitrogen and turn it into a carbon. And you know, but really it would be cool to do that in the way that we create music, right? It'd be cool to be sitting at like a big, you know, mixing board and turning the knobs that say, okay, I want the solubility of this drug to go up a little bit. I want this to be a little bit more potent against the you know, the enzyme that we're trying to target. And I want it to have like a little bit less exposure in the liver, because we're seeing toxicity there. And so, you know, just imagining that as a very tactile experience where you had like, you know, rather than going through 18 billion spreadsheets and, you know, kind of sort of understanding what you're doing, but like really what you want to do is you just want to tweedle the knobs and just say, all right, like I want to turn up this knob. I'm going to slide in a bit more, you know, like solubility and a bit less toxicity into my molecule. So we got a long way to go before we get there. We're having a lot of fun in the journey. And the other thing that we've learned is that musicians are latching onto this like you wouldn't believe. I mean, hopefully, you enjoyed our melody of vitamin B12. All these molecules sound great. We have a lot of different musical taste in our lab, so some people don't like certain songs, and some depending on like different cultural references, we can adjust the music accordingly. But imagine you're falling in love and you want to write a song about that moment, right? I mean, maybe you think about oxytocin that's circulating through your body as you're falling in love, and so you could type in oxytocin into our algorithm and get out a new melody, and that could then serve as an impetus for a song about that moment in your life. As one area that we're excited about.
Yeah. I was also thinking this might also be amenable to sort of unsupervised machine learning. Essentially, if you don't have labeled data and you have this musical notes or musical constructs, perhaps pattern finding on that data could be quite interesting, right? Even if you don't have any ingoing hypothesis, the machine could create some hypotheses from that type of data potentially.
There's no question. There is a huge body of machine learning and AI for generating music. And we were unaware of that when we began this project. We are deeply aware of it now, and there's a whole community of AI for music. Think about where AI starts, right? It's in image recognition because there's so many images on the internet, right? There's tons of music out there, right? So we talked earlier about how there's not much reaction data, or there's not much molecular data that at least isn't tied up in proprietary databases, but there's so much music out there that we can enjoy. So we're interested in how this paper will help us to bridge into. We've already been borrowing musical algorithms to develop new reactions, which we couldn't do before, but we're just completely unaware that these algorithms existed. You mentioned deep learning, and so this one of our earliest experiments was to train models on music, train models on molecules, and try and blend them together. That was very early, and we made a mess, and we had to take a step back. We're looking at it now, but we would get like I think we were anticipating that melodies would come, and they surely did. But the thing we didn't anticipate was like the breaks in music. So we would get like a 30-second musical register with like 2 notes. It would just be like beep, and then just like long pause for the longest time, and like some new notes. So there's a lot to learn in this space, and we're obviously we're having a lot of fun with it, and generating a lot of new connections and building new networks with making new friends in this space. So it's been great, but it's taught us a lot about the way we're thinking about drug discovery and reaction invention.
Yeah, I mean, it sounds a bit like the grand unification attempts in physics, Tim. So music is something fundamental. You're sort of using music to unify all sorts of classes of chemicals, because they will all foundationally could have a music. I mean, the same thing, string theory is sort of in that direction, to say there is something foundational to all the stuff that we see, and that might help us to understand it more deeply. And it's a really interesting direction. I don't know much about it, but it sounds quite exciting.
There's no right answer in this space, so we try not to have too many rules in this space. In organic chemistry, there's a lot of right and wrong answers, but in this particular space, there's not a lot of rules that we have to follow. So we're having fun. Excellent.
So yeah, thanks so much for spending time with me, Tim. This was great. Thank you.
It's my great pleasure. I have a 5-year-old daughter who I just recently introduced to the arcade while her mother was traveling, and she was playing a skee ball where you throw the balls into like a little hole and you win tickets that you can win a teddy bear.
Yeah.
I told her, I lied, that my wife is really, really good at skee ball. She's not, but she's good. She's good. But anyways, they just got home, and so my daughter's very excited to go see how good my wife is at skee ball, and hopefully win a big teddy bear. So that's what's on my docket now. Thank you so much for your giving me a chance to share our research.
Absolutely. Yeah, have fun. Thanks again.
Okay. Take care, Gill.
Bye.